used as a identification tool as a biolog system. 16S is one. So, I mean, these techniques are very much sound and available recently at our place in India. Anywhere using custom facility can be done. Like sequencing is very easy, one can get. Biolog system is custom-based facility somewhere is available. If you don't have biolog system, you can go for Burgess manual, which is in our library. One manual, which is actually a kind of listing the type of fermentation by each. Using carbohydrate fermentation pattern. Anyone want to ask anything else? Okay. If someone has joined from microbiology department?
Yes, sir.
So they have a demonstration from microbiology department, and you can ask that if someone is there, they have to demonstrate the laboratory instrument from the central instrumentation facility. We are going to connect with the microbiology department.
Hello, yes, sir.
Yes, sir.
Yes, Nitika, you are from microbiology department?
Yes, sir. Sir, we are.
So might be. Yes, you are not audible, Nitika.
Sir, we are operating the system. We will connect within few minutes.
Thank you so much.
Thank you, sir.
Anusri is asking, can we consider production of antimicrobial substances by starter as a primary or secondary identification factor? See, Anusri, it is like most of the bacteria, they produce antimicrobial compounds. So just based on the production of antimicrobial substances, we are not sure that it is probiotics, but probiotics generally produce bacteriocins and other antibiotics also that can be used as an identification mark, but not as a sole starter. As the beginning is not a sole feature for identification. Yes, it is a secondary identification system that we can say, but we cannot include as a primary. But it is a very important. Yes. See, non-probiotics or pathogenic bacteria also produce bacteriocins or antimicrobial compounds, but they are not probiotics. So production of antimicrobial compound is a kind of defense system for the strain. Defense protection by the producer strain, which kills the other strain. But if probiotic is producing antimicrobial compound, it's something which is a synergy in the feature. This is what I have taken, that one is gram staining, catalase negative and antimicrobial acid production. And if you test antimicrobial activity, that is great. Yes, anyone, anything? So this type of lecture, so you should interact to the best of your capability to make it possible that what idea we are having and tomorrow we are going to perform the experiment. So you are very lucky that you are exposed to with a very renounced personality from the US, and he is going to deliver his expertise in the last 50 years he has utilized. He has a very renounce in his area, and if you find his expertise, I mean, he's not only teaching you, but you can interact and you can ask and try to implement that idea in the laboratory. That is the real motive of this such type of course. That export from different part of the world is actually inspiring you to make it connected, to make it possible, which is not available here. Some of the idea, if you are reaching idea, you can make the world leader. If your idea is better, you are the leader. Now the scenario is changed. The money cannot make you leader, but if you have better idea, you can have a better work, better research. You can have better patent, better publication, good quality research, only with the ideas. So this is what the tutorial is made for. It's not listening, but whatever you have learned in yesterday lecture, you should interact. I mean, if I am not able to clarify you, I can put this same problem with the processing in a system. He will clarify things. Sir, can we check the growth inhibition of oncogenic bacteria with the help of probiotics? Yes, why not? We can check. Yes. And there are several reports you can find that probiotics are anti-cancerous. They inhibit the cancerous cells, and it like induces apoptosis in the cell, cancer cell. So one of the patent claims that probiotics induces apoptosis in the cancer cell only, but normal cell remains healthy. It doesn't target the normal cells, but targets the cancerous cells apoptosis. Apoptosis is something which is programmed death of the cells. So it induces programmed death in the cancer cell. Such a good agent. Yes, these probiotics produce antimicrobial compounds, and you can screen for oncogenic bacteria. So we are fortunate that Dr. K.K. Sharma, Head Department of Microbiology, he kindly agreed to deliver the demonstration from his laboratory. Actually, we are very thankful to him that the participants from this GIAN course are benefited. They have developed certain set of instrumentation technique, and if that one is useful for us, we will be connecting in a while, and we'll get.
So we are connected here.
Thank you. Thank you, Vicky.
Good
We are
afternoon.
expecting to. Yes. We are expecting you to demonstrate some of the instruments we are having, and all
Yes.
participants will be joining.
Good afternoon, everyone. I am Vicky from Department of Microbiology. Here representing our department. Our department have several grant funding from different funding agencies and the other grant commissions. But currently we are representing here the police channel, which is funded by DST Fish grant, and we have in this laboratory majorly we have the HPLC instrument. We have purchased from that funding agency, and we have also the GC instrument, which is also purchased by the funding agency. And demonstrate about the HPLC system. The HPLC system, HPLC is basically the high performance liquid chromatography which is used for the analysis of the liquid samples which are non-volatile, which can be interact with the column, and after the interaction of the column we can visualize the chromatograph in the form of peaks. These peaks can be quantified and identified on the basis of their retention time and the height of the peak. In this system, we have the quaternary system of the HPLC, in which here is the robotic head of the system, which take up the sample from the sample tray and put up into the injection port, and injection touch the samples and insert into the injecting loop. Here the mobile phase comes through using various pumps. There are majorly four pumps. These are our system is quaternary system. If the pump supplies the mobile phase to the column, the pump supplies mobile phase to the column. Here is the column. The column interact with our samples. The matrix show binding affinity to the samples. On the basis of the binding affinity of the sample, we will look out through the mobile phase, and the mobile phase pass through the detector, and the detector reads the analyte, and it will give the peak in the form of chromatograph. Here is the next one is the there are two types of detector. One is VWD detector, that is variable wavelength detector, and another is RID, refractive index detector. Both detectors are highly sophisticated and is up to date with the time. The RID is also known as the universal detector. VWD is the variable wavelength detector, which can be used for the different wavelength of the on the basis of the absorption maximum of the compound, which can be visualized. Here is the software system. We have the best software from the company Agilent supplied, and we have the system fully automatically, which can be run for overnight 24 hours continuously by supplying the continuous mobile phase. You can also check various compounds such as phenolics, purals, or sugars, proteins, amino acids on this, and other also organic compounds such as glycols, which are abundantly available in polluted water. Next, we will carry on to the GC system, which will carry forward by the RO.
Vicky, Vicky, can you hear me? I would like to start on the HPLC itself. Hello? Vicky,
yes, sir.
Yes. I would like to take a few minutes on the HPLC system. You know, HPLC is very important for probiotics also, because most of the probiotic bacteria secrete bacteriocins, which is a small peptide. And HPLC system, what Vicky is showing, is a very good system which efficiently resolve the peptides. And anyone who are having a mixture of peptide can use this HPLC system to purify it. If you are taking a purification part from the bacteriocins or sulfur supernatant, and you can grow the probiotics, most of the bacteriocins are secretory in nature. So if you are having a probiotic bacteria in your lab, you can grow and take the supernatant. Those supernatants are having several contents of bacteriocins in it, that is small peptide. And using a step-by-step process of purification, as Vicky suggested, that one can separate this peptide on the HPLC system. So this is what I wanted to add, Vicky, that this HPLC is very important tool for probiotic application, even for prebiotics. For example, if anyone want to isolate prebiotics from plant extract, this is a very important system. One can go for the column specifically for the carbohydrate oligosaccharides. and separate these prebiotics. So using HPLC, you can purify the prebiotics from the mixture, and those prebiotics can be used for the growth of probiotics. Yes, Vicky, please continue.
HPLC is a very highly sensitive technique. By using that, we can calculate 0.1 ppm of the concentration of by using the HPLC, and we also can use separate column for the separate bacteriocins and small peptides, which can be identified as well as can quantify by using the HPLC. Now we further move for the GC system. The GC will be demonstrated by the Arush. Good afternoon, everyone. Myself Arush, and will be carrying forward with the instrument data. So this is our department health provider service system. In case you want to analyze volatile sample, you will go for the GC gas chromatograph. So we are equipped with chromatograph and two types of detectors, which are FID and LPD. FID is flame analysis detector, while LPD is nitrogen phosphorus detector. So the principle is we have three types of gases. One is the nitrogen gas, which is the GIAN gas. So these three cylinders carry three different types of gases. This is the nitrogen gas, which will be used as the carrier gas. That is, it will carry our sample through the column. Second, we have hydrogen, and it is used to fuel the detector. And third is the zero air. Because hydrogen and zero air combine, they will undergo the process of combustion, generating a flame, and that flame will be used to detect our sample. So after the carrier gases have passed through the cylinder, here we have three traps. These traps are used to further purify our gas so that it is free from any impurity. So that the gas which is passing on to the instrument is free from any impurity, so that it do not degrade our column. Our GC instrument is equipped with photo sampler, so you just need to pass the command to the system, and it will take up the wire that is at a specific location that you have mentioned. Take up that wire and move into the aeroplane. And here is the syringe, tapped into inlet, it converts into volatile form and moves into the column. This whole setup is called the instrumentation oven. In the oven, we have our column, and through the visible, but there is an inlet pipeline, and through that inlet attached sample will move to the column and get into the detector. Here. So after the column is attached to the detector, these two are different types of detectors. One is the LPD, second is the FID. In case you want to analyze organic compounds, you can go for FID, which is flame ionization detector. And in case you want to analyze nitrogen or phosphorus containing compounds, such as halide or pesticides, you can go for LPD detector. After the compound is analyzed, the electrons that are released will be shown in the form of a heat peak. And these peaks can be used to find the retention time, and based on the retention time, we can check where is our compound and how it is separated from other compounds. And the peak height or peak area denotes the intensity of the compound. Thank you.
So Vicky, this is completed or anything else from your side?
Yes, sir. We said and yes,
Any other instrument you want to demonstrate?
we are conducting a workshop of these students, and these are the participants that have joined the workshop.
Hello. Good afternoon. As we have the office funded lab, we have two highly analytical instruments in our laboratory, that is GIAN and we are successfully organizing the workshop for the training of the participants from the different regions of the country. And we have successfully completed the two workshops and trained more than 25 participants. And we have planned currently the third workshop is going in our department. And these are the participants from the current workshop. The next workshop will be planned in the month of February. So it is for the participants of the GIAN program, so who want to get training about the extension GT can join us in the month of February. Thank you.
Thank you, Vicky. I would like to add that this is a facility run by Department of Microbiology, and they have a very high-end instrumentation facility which helps not only in the probiotics but different sections of microbiological experiment. And if any one of you on this GIAN portal want to get hands-on training or want to get your sample done here, these students are very active and they are dedicated to handle these instruments. And the credit goes to head of department, Dr. K.K. Sharma, for organizing such a activity. So this is the reason I connected with this laboratory, that it is one of the most functional laboratory in our university, which is providing a best platform for the students to get hands-on training. So you can not only think of, but connect with this, and at a very nominal cost, these students are helping, head of department is helping to get the training from this instrument, which probably you are not able to have in your laboratory. So if you don't have these instruments in your lab, you can come here and register for a nominal fees, and for a week or so they organize this program, and most of the technique related with the microbiology or micro handling to the product handling technique can be learned. It seems that these are the small techniques, sometimes, gel electrophoresis or isolation of DNA. But this is a very important, whether you are isolating DNA in one of the most purest form or not. It is not only isolation of DNA, but isolation in the most purest form. Same way with the electrophoresis, if you run the electrophoresis in a most technically sound manner and learning with the technicians, then your experiment is better than the others ones, right? Resolution of clear band, because all these things are required publishing a paper. If you submit a paper with a good quality gel image, that is very much acceptable. If you are submitting a paper is a very blur or very poor type of images, and the resolution is not clear, then it is not accepted by good quality international publication. So I congratulate the whole team of microbiologists laboratory for you, and they are involved in the demonstration of these equipments along with the head of department. And I thought that this is very beneficial for our GIAN program participants. So any one of you who are interested, you can write to me or head of department microbiology. Information is available on the website. You can write directly to HOD microbiology and find this instrumentation facility useful. And thank you very much for the entire team for presenting your instruments and also explaining the things with the GIAN participants. Thank you very much.
Thank you, sir. Thank you for giving us this opportunity. Thank you, sir.
Anyone from the participant want to ask any question with the team? You are most welcome. Still Vicky is connected and he can answer if you want.
Video answer.
No one is asking, so okay, from microbiology, it is very good that you did. We are already done. Yes, one is asking that how much concentration is required for HPLC. It's a very sensitive. You can use like in the nanogram protein that is detectable on the HPLC. Minimum you need something like 2 microgram protein which will be detected on the HPLC to give a very clear peak.
Hello.
Yes, Vicky.
The question from the chat box is that the minimum required of the sample for the that is the we required only 300 microliter of the sample for the
That I clarified not only in the terms of volume, but you need minimum 2 to 5 microgram protein in 100 microliter sample that can be easily detected.
Any question from any other one?
Vicky, one question, they are using the what kind of gases you are using in GC?
Sir, we are using nitrogen, zero air, and hydrogen.
Okay, right. Any other question? Anyone want to interact with the microbiology laboratory? Okay, Vicky, I think there is no question, so thank you again. How much cost for one sample analysis for HPLC? One student is asking,
Sir,
what
it
is the
depends.
cost?
Sir, it depends upon the sample quality and the sample system. If he is providing the mobile pill, then it is 400 per sample. If we have to purchase the mobile for that sample, it is 800 per sample.
So it's a very, very nominal.
Yes.
At a very nominal rate, you can learn, you can be trained, and you can do your sample also. It's all right. If there is no question, Vicky, thank you for answering the questions from the participants.
Thank you, sir.
So this is what I wanted to demonstrate you this instrumentation laboratory, where not only you can do your sample, but you can come direct and get training of these instruments. I mean, don't think of small instrument or large instrument, but hands-on training is very important. Actually, GIAN program is organized for only for that purpose, that how a person can be trained and like electrophoresis or HPLC or column chromatography. That is one of the part, but handling the instruments correctly and getting this result accurately is a very important aspect while performing the experiment. And rest of the laboratory demonstration we will do in our laboratory. Most probably it is tomorrow. So tomorrow we'll have some interaction in our lab with the different instruments which are actually used for right from the growth, identification, purification of metabolites from the probiotic bacteria and their identification, their characterization. These instruments will be visited tomorrow. So if there is no question, I think this is the only aspect I wanted to cover it today in the tutorial section. If it is fine, it's okay. Otherwise, you can interact with me. If anyone want to interact or ask question, you can talk to directly. You can unmute. This is open. I think you can mute. Yes.
So I had asked one question, like how this prebiotic selectively help in the growth of probiotic microorganisms in the GI system.
Can you repeat, Chandni, your question again?
Sir, how this prebiotics selectively help in the growth of probiotic microorganisms in the GI system?
Yeah. In the GI system, we have a gut microbiota. We have a microbiota, So there may be probiotic and non-probiotic both are present there. And if you are giving a nutrition which supports the growth of both probiotic and non-probiotic, then it is not good for us. We have to selectively support the growth of probiotics only, where the pathogens should be killed. There are two mechanisms. Number one, if I provide a prebiotics, the pathogen will not able to utilize those prebiotics and will not grow. Number two, if you are using this prebiotic, the probiotics will utilize the prebiotics and probiotics will be able to multiply at a faster rate as compared to without prebiotics. And if you see other aspect, the probiotics also secretes antimicrobial compounds such as lactic acid, bacteriocins, and these bacteriocins inhibit the growth of pathogenic bacteria. So what I am doing, I am providing a prebiotic to the gut, and these prebiotics helps the probiotic to grow and secrete the antimicrobial compound like bacteriocins. Those bacteriocins are killing the pathogenic bacteria. So it helps the selective growth of probiotic only, and other bacteria are being killed. I hope I answered.
Yeah, okay, sir. Okay, thank you, sir.
Yes. This is very important question, thank you for asking, because such type of clarity using prebiotics would be there, that use of prebiotics is supporting the proliferation of probiotic bacteria, and indirectly it is killing the pathogens or non-probiotic bacteria there. And using prebiotic, it is a nutrition. If you are using this nutrition, they are more proliferating, more growing. So their adsorbent capacity, their adhering capacity, their co-aggregation facility will also be higher, and they will be adsorbed more on the surface of epithelial cells, will be aggregating and forming a layer over epithelium, performing the function of probiotics.
Sir?
Yes.
So this prebiotics will also help the growth of other beneficial microorganisms also, right? Except this probiotics too.
Yes, it will help all the probiotics to grow.
Matlab non-probiotic beneficial microorganisms that are in the gut, they also will be grown in the presence of this prebiotics, right?
Generally, non-probiotic bacteria are not able to utilize prebiotics.
Okay.
So only it is considered that the probiotics are utilizing these fructooligosaccharides.
Okay, so thank you.
This is the reason you are having.
In addition, one more thing is that probiotics, the byproduct of these prebiotics is leads to the reduce of the pH, and this in turn also facilitate the growth of probiotics only.
Okay.
So this is one thing that selectively increase the growth of probiotics only. Like inulin and other prebiotics, their byproduct leads to the reduction in their pH, which in turn enhance the growth of probiotics only, and reduce the growth of other pathogenic bacteria. Other pathogenic bacteria are not growing. Sorry.
But it is being said that these probiotics are non-digestible components, right?
Yes, right.
So how it is getting broken down in the beginning itself in order to reduce the pH?
Okay, human body can't digest this, but bacteria can degrade these prebiotics in their simpler form to get their benefits. As can use as
Okay, so this degradation is done by probiotics or other microorganisms at the beginning.
Let me clarify.
Okay, sir.
The prebiotics are degraded by, I would say, like consumed, utilized by probiotics only, not by other bacteria. So by doing so, it allows the growth of only probiotic bacteria. And if the probiotic bacteria are growing, proliferating faster, they create an environment which is acidic. And if the environment remains acidic, the other pathogenic bacteria are not able to grow in that environment. And basically, that ecological system allows the proliferation of probiotic selectively. Chandni, hope it is clear?
Yeah, yes, sir. Thank you, sir.
Yeah, and those prebiotics are not by which can degrade the prebiotics, like what Pallavi is saying, inulin, phos and oligosaccharides are not being degraded, so they are as such removed by the GIT tract. Okay, then if there is no question, I will stop this session here only, and we will be connecting at 5:30 in the evening today. I will request all of you to join at least 15 minutes in advance, so that our expert will join at 5:30, and we'll start the session after he joining. Thank you very much for all your attention and asking the question. The tips for the tomorrow session is that you should also join at least 15 minutes before, so that you should ready at 11, and keep yourself interactive. Whenever you ask in the session, come with the questions, come with the ideas, and that idea should be doable. It's not should be only for discussion purpose, but also whether we can utilize such type of idea for our work, for our experiment, how to improve it. That kind of things should be included as a part of discussion in this tutorial section. All right, so have a nice day. We'll see you in the evening 5:30.